So we've been talking this morning about advanced therapies, and I think it's fair to say that the majority of the advanced therapies that have come into the clinic and have gone into commercialization over the past decade or so have been a result of advances in cellular engineering. And MaxCyte's focused on the basics of cell engineering, and what we're going to do today is kind of lift the bonnet, lift the hood about this whole area of cell therapy and cell engineering, and talk about a technology that MaxCyte's pushing forward. And then also we're going to talk about, we think, is a brand new way, a breakthrough therapy in the CAR space for using the transfusion medicine infrastructure around the globe to deliver CAR therapies quicker, less expensive, and to solid tumors. So we just went public on the AIM stock exchange, so please read through that. The company is focused on a cell engineering platform, as I mentioned, and we're focused on drug discovery, making major advances in drug biologics development, and also in cell-based therapy, again, engineered cells. This company has very quickly established itself with over 50 of the top pharma and biotech companies as clients, 9 of the top 10 around the globe, the top 3 in Japan. And we also license our technology to developers of cell therapy products, and we have over 35 programs with companies and universities who use our technology to develop cell therapy products. I mentioned we are also developing our own product called Karma. It's a platform for using fresh cells. And I also want to talk a little bit about a convergence we're seeing, and that's the convergence between immuno-oncology and gene editing, and MaxCyte's right in the center of that. We had a successful AIM offering in March of 2016, rather difficult time to go out. Our friends at Pam Muir-Gordon took us out, and Concilium Strategic Communications helped us with that effort. We raised about £10 million with blue chip investors, including BlackRock and Legal & General. So we've got a great institutional shareholder base here in London. So our highlights in 2016, we were able to establish a far-reaching strategic relationship with Johns Hopkins University in Baltimore, one of the leading groups in the world in oncology care, and I'll talk about that toward the end of the discussion. I mentioned over 35 programs where companies are using our technology in cell therapy, and as these programs move from phase 1 to phase 2 to phase 3 and eventual commercialization, that value to MaxCyte increases rather dramatically, and we'll talk about what that looks like. We are a public reporting company, so in the first half we reported sales of $5.5 million, which was a 30% increase over the same period last year, and we had a cash balance of slightly over $12 million at the end of the half year. So the business is in 3 major verticals. This is our business model. In the left-hand side, you see us selling our systems into drug discovery. Again, this is with the big pharmas and big biotechs in Japan, and you can see some of these names are highly recognizable, who use our technology for, again, small molecule drug discovery using cells, and also using cells to rapidly produce novel biologics. The center is where we license our technology to cell therapy developers, and this is where we receive license fees and milestones from these companies. And as they move to the clinic and they move through the clinic into commercialization, MaxCyte's very well poised to share in that value as they move and commercialize. And we have focused our attention on commercializing this technology. And in fact, we have our one commercial partner in Japan who has commercialized a cell-based therapy using MaxCyte's technology in 5 centers throughout that country. And in our Karma program, we work with the top groups in the CAR field. We work at the University of Pennsylvania, been noted here, National University of Singapore, which is Dario Campana, who is the scientific founder of Juno, and with Johns Hopkins University in Karma. And Karma really is a breakthrough in immunotherapy. It's, again, using the transfusion medicine infrastructure, freshly isolated cells loading with messenger RNA to express CAR molecules to treat solid tumors. So what is this? What's behind all this? Well, it's called flow electroporation. This is a crowd that understands transfection, so I won't go through the benefits of electroporation other than the fact that when you probably used it in grad school, you probably killed some cells. Well, that's not the case with this technology. We have created this technology purely for clinical purposes. So over 90% of the cells that we process using our technology are engineered, and over 90% of the cells are viable, so they go back into the patient. We do this in a closed environment in a fully integrated system. So again, it can be incorporated either in a large-scale manufacturing operation, or it can be deployed in multiple locations around the world. And why do you want to engineer cells? Well, there are tens of thousands of pathways that scientists have been able to identify that one would like to be able to either upregulate or downregulate in some way to change that cell. To change that cell to discover drugs, that cell can be used as a substrate, so it lights up if a certain small molecule hits it. It can be used to produce proteins. Either they be standard monoclonals or multivalent antibodies. Pretty much whatever the imagination of the protein expression scientist has, we can use our technology to express that cell in production cell lines. You can use the cells to redirect the cells to attack cancer cells. And that's the basis for immunotherapy, whether it be dendritic cells, T cells, NK cells. Whatever the cell type is, MaxCyte is working in that cell type to redirect that cell to go after cancer. And finally, you can actually, and we all know this, edit and correct genes. Either knock out a gene or correct a gene in either a stem cell or a T cell, and MaxCyte is working in that area. So those are the major areas where a cell engineering technology is critical for making a major difference in creating new drugs. In drug discovery, we work with companies to identify novel drugs based on cells. We work with, for instance, AstraZeneca, who have published that we've helped them to identify novel psychiatric drugs that they could not identify without our technology. We've worked with leading manufacturers of proteins in helping them to dramatically shorten their development time. We've also been able to help companies develop products that couldn't be produced without our technology. That also includes AAV vectors and lentivectors and recombinant vaccines. And I'll talk about that in a few minutes. And then finally, cell therapy, where we work with companies this is a non-viral method, so we reduce risk. We also decrease toxicity. We can improve potency. And because this technology, again, be deployed in the laboratory, we can take it from the lab all the way into the clinic, then eventually commercialization on the same exact platform all the way through. In drug discovery, we sell our instruments. We sell two instruments. One is a small-scale instrument that runs 10 to the 10 cells, 100 milliliters of cells in about 20 minutes. That's the upper right-hand corner. And what you notice up there is a blood bag. That's a 100 milliliter blood bag that is used for our cells. So that's the output of an apheresis instrument. It's also the input of our instrument. So this is a highly interconnected system for virtually any manufacturing process that's required. We also have a large-scale system, which we call the VLX, which runs a liter of cells. That's the output of a 200-liter bioreactor in about 20 minutes. And companies that work with us are using this technology to produce novel vaccines for Zika virus, for Ebola, where the vector itself is toxic to the cell, so they have to do it in transient production methods using our technology. Companies are also using this technology to produce large-scale lentivector in suspension cells and also AAV vectors in suspension cells. In the cell therapy partnering area, this is an area that obviously ARM is focused intently on, and there's been a tremendous amount of investment in this space. That's why you're all here, of course. But our technology has been being used in helping companies move their programs along. And as you can see, in 2013, we had about a dozen programs, including groups like Sangamo, who use our technology in their ex vivo programs. And we reported in the middle of 2016, 35 programs. So there's been an explosion of these in the marketplace, and MaxCyte is well positioned, not only in the clinic, but as, again, these programs become commercial, MaxCyte will work with these companies to be able to commercialize these therapies as they move forward. And one of the reasons, obviously, why there's been so much investment in this space, as reported by ARM, is because of the breadth of the indications. We're working in oncology. About half of our programs are oncology, but they're also in genetic inherited diseases. They're in cardiovascular disease. They're in diabetes. Pretty much anywhere where these scientists can dream up a therapeutic context, and we can move these cells into the clinic and help them through their therapeutic area. And finally, Carmen, I wanted to kind of breeze through the rest here so I can spend most of my time talking about this program. So we all understand in this room the power of the CAR T space. And I think it's hard to overestimate how important this has been for the industry and for patients. Over 200 patients have been treated, really just phenomenal results. And it's brought in quite a bit of investment, of course. The companies listed here all have a combined market cap of over $10 billion. A few years ago, they had a combined market cap of maybe $2 billion. So this area has really, really moved dramatically and is extremely important. But there are limitations, and that's why we got involved in this area early. We've been involved in the CAR field for 8 years now with two of the leaders, Carl June and Dario Campana. And the issues are, one is this is certainly limited to certain blood cancers. I think that's well aware. And it's also, there's this on-target, off-tumor toxicity. And with CD19 is indiscriminate. It goes after leukemic cells. It also goes after normal B cells. So every one of these patients has B cell aplasia. Can be managed with IVIG, but again, it has to be managed. The other issue is that it's an expensive manufacturing process. We've talked about that, can anywhere from 5 days to over 15 days. And going back to the toxicity, when researchers have tried to use this approach to solid tumors, they've had SAEs and actually patient deaths. So it's very difficult to control these lenti-vector-based CAR therapies, because once you give that therapy, the cells expand, and it's very hard to control the outcome of those. There's been a lot of work that's being done, investments in switches and rheostats, ways to cut back on these CAR therapies in the future. But our approach is a little bit different. And what we were asked to do by both Carl June and Dario Campana was to use our expertise in messenger RNA, loading messenger RNA into cells. And when you put messenger RNA into cells, the cells will express the protein for as long as the mRNA is inside the cell. And that takes anywhere from sometimes 3 to 5 to 7 days. So you have now a T cell that expresses a CAR molecule for 3 to 5 to 7 days, and then it automatically, well, not automatically, it dissipates and it goes back to become a normal T cell. And what we're doing at University of Pennsylvania right now, National University of Singapore, is there's 7 clinical trials going on right now. Most of them are in solid tumors using our technology, loading messenger RNA into T cells, expanded T cells, and then going after solid tumor indications. And that includes pancreatic cancer, it includes breast cancer, glioblastoma. And we're starting to see some reports back from these locations at ASCO and AACR. So what we're seeing is it's a safe process, and we're starting to see some anti-tumor activity. So what we did at MaxCyte is we realized that if you're taking lentivector, the cells have to be in expansion phase. They have to be in cycle for the virus actually to infect the cell. But with electroporation, you don't need those cells to be in cycle. And so we did the experiment. We said, if you were to take cells that had been expanded and then cells that are fresh, and you run them both through our MaxCyte system, we were pleased to find that both of those cells behave the same way. So freshly isolated cells actually have the same anti-tumor effect, and perhaps even a stronger anti-tumor effect than selected and expanded cells that are currently being developed for CAR therapy. So we took that and we developed a process around that. We have issued patents around placing mRNA into fresh-slated cells to treat a number of different diseases. And what we're doing here is we're leveraging the existing infrastructure of transfusion medicine. You heard about this morning. There's over 20 million procedures done in transfusion medicine a year. They know how to do this. They're in virtually every hospital, including community hospitals and oncology centers. And what we're doing here is it's a very simple process. You take an apheresis from a patient, you do a minor cleanup, no selection, just a minor cleanup. You run them through the MaxCyte technology with messenger RNA. Then you have enough doses at that point to redose the patient between 6 and 8 times. So although the process, and it takes less than a day, so although the cells only last and express the CAR molecule for 3 to 5 to 7 days, you have enough doses frozen to give back to the patient on a weekly or biweekly basis for several months. So we think this is a major shift in the way to think about giving CAR-based therapies. And what we're also doing is we're focusing this on solid tumors. So we have a relationship with Johns Hopkins, and at Johns Hopkins we're completing our preclinical studies. We raised the money through AIM in order to support a clinical study which will kick off in 2017. That clinical study will include platinum-resistant ovarian cancer patients, where we believe we have a pretty good shot at showing an anti-tumor effect. And then once we do that, we have a platform. Again, the platform is a fresh isolated cell, and use that as the basis for the next generation of cell-based therapies. So I want to thank you for your attention and look forward to talking with you about MaxCyte and Karma during the meeting. So thank you.